afternoon to all. Greetings on this Independence Day. Thanks to Professor Srinivas Chakravarti for arranging this nice, lab meetings. Today we'll be talking about the peripheral neuroscience computational modeling of the Pacinion Corpuscle. It's a summary of our lab projects. Couple of papers what we recently published. A special focus on the tactile perception. Before I talk about the summary, I want to briefly introduce our lab. It is in the Department of Applied Mechanics. The department has three major different groups. Biomedical Engineering, Solid Mechanics, and Fluid Mechanics is considered as one of the most interdisciplinary department in the institute. Biomedical Engineering is one of the three groups. It has a very long history. Way back when 1961 started by one of the NASA returning scientists. First one in India along with the IIT Delhi. Touch Lab is more recently established, 2005, about 15 years ago, when I joined the institute. The main focus of the Touch Lab is to understand how our touch sensation is working and then develop applications, technology to give touch feedback. The focus is on the translation research. Although we do a lot of basic science, but also we want to focus on the translation research. We have 4 startup companies by our students, 2 more in the pipeline. One of our notable contribution is by our first MS scholar, Dr. Hari Vasudevan. He went on to Apple and then designed this taptic engine. Yeah, latest feature available in the latest Apple cell phones. We are very glad that now we have a contribution to a global product. Another notable contribution from our lab is by our Dr. Abhijit Biswas, a PhD scholar from our lab, is joined the IIC as a faculty. Now coming to our today's topic, what do we model as far as the touch is concerned? Why touch is there and what is there to model? Not to explain, I need to explain to you some applications and then I'll go into the modeling. Today, VR, AR, MR, these are all, a lot of industries are talking, a lot of startup companies are working on it. One of the grand challenges in virtual reality is that there is no touch feedback in the virtual reality. We wanted to touch the virtual object. Providing touch feedback in virtual reality is one of the grandest challenges. So in the last 15 years in our lab, we have been solving this particular problem, but focusing on one particular specific target applications, helping the clinical doctors with medical training simulators. Both our lab and the startup company, Medical Haptics, together, we have created about some 10 medical simulators and then putting in use with the doctors. The first one was the epithelial injection simulation. Those of you are aware of what is epithelial injection. It's a very, very risky procedure the labor women take. If there's a small mistake in the needle insertion, the subject or the patient will have a lifetime damage to the neural system. So far, the doctors have been practicing on the patient, putting the patients on risk. We have created this simulator where the patient doesn't have to be there, only we can practice on the computer with a realistic feedback. The doctors can practice as many times, kill the virtual patients as many times, and then get the skill, thereby improving the patient safety. There are other simulators, which I am not going to the details. If you are interested, now we can visit our website for more detail. One particular simulator I wanted to highlight here is the in vitro fertilization training simulator. The IVF procedure needs very fine motor skills. We need to apply right amount of force at the right time, at the right place. If you don't apply, the whole procedure becomes a failure. Now we know why IVF has a very high failure rate. Again, the doctors practice on the patients. They damage about 30 patients before they become competent. If you are one of those first 30 patients, then the risk is very high. So again, we made a simulator for the entire IVF procedure in virtual reality with a haptics device feedback. The technology has been transferred to a Singapore company, and the Singapore company is now working with a Finland company for 13.5 million euros. We are very glad that this product is reaching the global market now. Another interesting application, recent application coming up is this is 5G. It is also called the tactile internet. It is called a tactile internet because it promises 1 millisecond latency. We are going to see that for haptics, we need this 1 millisecond latency. Otherwise, we can't do haptic rendering. So this 5G internet enables lot many new applications where touch feedback can be given. The Zoom meeting we are talking about, or the Google meeting we are talking about, now we can only see and hear things. What if we can touch the slides or touch the objects in what you see in the Google Meet? That is going to be the feature in the 5 years down the line. For this to happen, we need to have a very good understanding of how our touch sensation is working, and therefore that will enable us to develop gadgets. But in order to understand, there are a lot of data needed. There are a lot of neuroscience experiments have been needed. It's already been done, but in our lab we don't do it. There are perceptual experiments needed. In order to understand the mechanisms, we also need neuroscience models to explain the neuroscience experiments. We also need perceptual models to explain the perceptual experiments. This all the neuroscience models will have help from the perceptual models, perceptual experiments, or neuroscience experiments. Neuroscience models will be validated by other three components. Similarly, perceptual models will be validated by the neuroscience models. It is all interdependent. So in our lab, we do the three components, not the neuroscience experiments. We collaborate with other researchers who have facilities in the facilities for doing neuroscience experiments. So our focus in the lab is specifically on the one of the mechanoreceptors. We are going to see what is the mechanoreceptors and what is the specialty of this one mechanoreceptor and why it is so special and why do we model it and what is the interesting applications we can develop from this modeling of one of the mechanoreceptors called Pacinion Corpuscle. Let me start with our introduction on the touch. Sense of touch is we call as one of the fundamental senses of human. Although we have 26 different senses, all these 26 different senses can be classified into 5 major senses. Among these 5 major senses, we call touch as a fundamental sense because the other 4 senses depend on this fundamental sense. Without this fundamental sense, other senses cannot exist. That is what our claim is. For example, we have a paper called on the somato sensation basis of vision. Vision cannot exist without touch. That is our claim. If you are interested, please take a look at it. The touch sense is by mostly by four major mechanoreceptors: Meckel, Meissner, and Ruffini and Pacinion corpuscle. But all these mechanoreceptors, which converts the stimulus into the electrical impulses, they act almost like a no bandpass filter. That is what our understanding is. Each sensor is filtering certain characteristic of the stimulus and then conveying it to the brain. Some sensors are the shallow near the skin. Some sensors are very deep. Some sensors are slow adaptive. Some sensors are rapidly adaptive, depending upon whether you want to capture the static nature or the dynamic nature. That is all very well known in the text. Right? Among this, PC we consider is the most sensitive mechanoreceptor. It functions as a cluster of two or three, not more than two or three. However, human vibrotactile sensation, because of this vibration, because of the PC bunch, it is very, very less utilized and then less studied as well. We have few neurophysiological data and then not so good no, computational model. In order to understand this PC very well, we need a very good computational model. So not only isolated models of the PC, but also the clusters models. Our focus is on modeling the neuroscience aspect of the PC, perceptual aspect of the PC, and then perceptual modeling of the PC experiment as well as the model of the human vibrotactile sensation. PC is responsible for the vibrotactile sensation. Again, this is a very well-known textbook stuff. The PC has an onion-like structure. It has 30 to 60 lamellae, we call it as onion layers. So each layer filters the mechanical vibration, and then finally the stimulus reaches the core, which is converted into electrical impulses. The total length is about 1 mm to 4 mm. Okay? So for this very important PC, there was a model by Lawrenstein and Scala way back in 1966. After this, there was no model at all. People have been publishing, phenomenological data, phenomenological model, but not no the neurophysiological model, neuroscience scientific model. Until we looked at it 5 years back in 2015. Our model is known as the BMS model in the literature. So, what enabled us to succeed? There were a lot of new physical data, physiological data coming up only in the recently. Then, after we published, some of the groups around the world followed our work and then extended our work to many other situations. We are going to see the details. So, from 1966 to 2015, what was the change? In the 1966 model, the core, the inner core, was considered as rigid, whereas we consider this as a viscoelastic nature. So only recently, the core itself is considered able to have the stretch activated ion channels. That gave us the hint that it has to be a viscoelastic material. And then the Young's model was assumed to be very very high, something like a very stiff like blood vessels. But we found out that that is inaccurate. Again, our recent data shows that it has to be around 1 kilopascal, which is equal to the cell stiffness. This is again validated by another group. What we assume this, and then went ahead and modeled it, and then got the results. Our results have been validated by another group in Minnesota. Again, the mass of the lamellae was not considered, whereas we had to consider it because we wanted to look at how this PC, in corpuscle, is responding to a variety of vibration, not just from 10 hertz to 1 kilohertz, even beyond 1 kilohertz, 3000 hertz. We wanted to look at it. This was the model, BMS model of a single PC. The top figure shows only the core. The green shell is the inner core, and the blue one is the neurite. The violet one is the neurite, axonema, right? And then the pink one on the right side. These are all the first Ranvier node. Equivalent electrical circuit have been created. And then using a simple integrated fire model, we could generate all the spike rate, and the spike rate characteristics have been validated with experiments. We have also generated a threshold characteristics, which was not there before at all. This was the first time a threshold characteristics have been created, and this was having a lot of practical applications to the extent that we could explain so many variations in the this curve, threshold characteristic curve, from the experiments from different groups. There are about 15 to 20 groups have different tuning curves for PC or human vibrotactile sensation. So we could explain using the model why there is a variation. This is one of the very beautiful thing about the modeling. The modeling gives us an understanding of insight of how the things are working, why there is a variation, right? So that model is only for one PC, whereas to be practical, we needed a cluster of PC. We extended this model into a cluster of PC. So a bunch will have 2 or 3 PCs, and from 2 or 3, the signal is coming out to be the same. The co-activation of 2 PCs units by a vibrotactile stimuli, shown like this, it is found to be the same. It is not two different signal. So this has led us to model a cluster PC. So in the picture you can see that the middle, the solid line shows the where the stimulus is there. The dotted lines, green and red, the dotted lines are the location at which the 2 different PCs are there. And each PC has a Gaussian receptor field. The mean and variance of the Gaussian receptor field tells you where the location and the responsive receptor field. The difference in the mean of the 2 Gaussian represents the distance between the PCs within the cluster. Now we have varied this distance between these two clusters and the location of the stimulus between these clusters. and we got this again, and the same the graphs. We used a very well-known communication algorithm called time division multiplication. How signals from two different species are multiplexed and then connected to the brain. We generated the spike rate characteristics and the threshold characteristics again, like for a single PC. We found that the spike rate characteristics is no different from the single PC. It's almost the same, whereas threshold characteristics were different. This again has a lot of practical use. To make it even more practical, we had to add one more feature, which is very important. People have been using electrotactile displays. What we were looking at only the vibrotactile, which is a mechanical stimulus. What we needed is also is the electrical stimulus. So we went upon now modeling the PC for electrotactile stimulation. Unfortunately, there was no model. Fortunately or unfortunately, there was no model at all. This was the first model we have created, and this is our recent paper there. So here, the PC is assumed to be down deep below the dermis, and two electrodes are attached at the skin at a distance. So we have this electrical equivalent of the entire model. The top portion shows the human skin interface, electrode interface. The middle shows the neurite, and the below shows the first triangular node. Again, we have generated the spike rate characteristic and the threshold characteristics. Now the spike rate characteristics are now entirely different than the mechanical one. So far people have been only having the experimental results. We could match again with the experimental results, and then infer many other parameters how to control all the voltage required to elicit the sensation. Then to improve the practical use of all these models, we have done something unusual, which we didn't expect it, but because we have got some insight from the model, we got this idea of why not we mix both mechanical and electrical hybrid stimuli, we call it as. In that case, what happens? Spike rate characteristics and the threshold characteristics. So we kept one mechanical stimulus constant and then change the electrical stimulus at different amplitude. And we found out the spike rate characteristic and the threshold characteristic. Similarly, we did the vice versa, kept the electrical constant, mechanical varied. This is again, it's not possible without the modeling. Just with the experiment, it is not possible at all to get this kind of insights. How this threshold characteristics and these spike rate characteristics are different from single PC or cluster PC or single type of a stimuli. So we could get some patents out of this, and then we did some demonstrations, and we are giving this technology to a Canadian company to put it in use along with their devices. So the inferences from our models are like this. All the spike rate and threshold characteristics are improved to match the experimental data very closely, and then we could predict some of the spike rate characteristic and threshold characteristics which is not there in the experiment. The practical implementation of these characteristics in the design of the tactile displays, both mechanical as well as electrical displays. How the behavior of the PCs change in combination with other modalities of stimulus is yet to be done. In the future, we are going to do. For example, how for thermal stimulation this PC is going to behave, or thermal receptors are going to behave. Right? Extension of our physiological models into understanding of the perceptual illusions. There's a perceptual models, right? There are very many illusions we wanted to simulate it, such as the Cochineal rabbit, thermal grill, and other things. There is going to be a feature. With this very short introduction, let me acknowledge all our collaborators. As, most of the slides what I have shown is from Dr. Abhijit Biswas, who is a faculty in IIC Bangalore, and currently Madan Kumar is working in our lab for his PhD. With this, I thank you again. I'll be happy to take some questions. Uh, um, uh, One of the questions from the audience is this. Let me try to explain it. The question is that why do we need elaborate models such as in our work? Why can't a simple model is used to explain the experimental results? More specifically, why can't the mechanoreceptors be modeled as second order filter? Would it be accurate? That's the question. The answer is yes, it can be accurate, but it will not serve the purpose. So the elaborate models lead to insight how the object that we are modeling is actually working. In our case, it's a PC. So the whole purpose of modeling the PC is to get insight into it. If you simplify with a second order filter, then it's now defies the purpose for which the modeling is done. We don't get any insight at all, or the insight we get from second order filter may be very limited. If we hadn't modeled with a elaborate model like this, we wouldn't have got the idea of extension, idea of the patents. These are all have practical implications. Therefore, even if the second order filter works accurately, represents the experimental data of the PC, it may not be useful to take advantage of the knowledge about the PC. Our interest is on the advancement of the knowledge about PC. Therefore, or we don't have to worry about whether it is a second order filter or not. We wouldn't have got so many ideas for practical applications had we used the black box models. I call this filter box models, filter second order filter model as a black box models. Right? Even the AI models, all the artificial intelligence models, they are all black box models. These black box models may have advantage in some places, but not in our case. Not in many other cases as well. So you need to know where to use this black box model, where you should not use the black box model. I will stop here. Thank you.